Hello, good morning, everyone. My name is Payal, and today we'll be doing our fourth session of Let's Build Research Aptitude for Interviews. And today we'll be discussing about Agarose gel electrophoresis. So, Agarose gel electrophoresis is a very basic technique, and the DNA which we isolated hypothetically in our first session, uh, now we are going to process it with the using the Agarose gel electrophoresis technique. So the first and foremost question is that uh, why to use and when to use this technique, and what is the working principle behind this technique. So first we'll answer this question, why to use and when to use. So like we do have DNA isolated in our hand. What we did, we just took the DNA, isolated it from plant, animal, or bacterial or yeast cell. Then we stored it in solution form by dissolving it in TE buffer. So now what we'll do, we want to check the quality of our DNA. So we checked its quality by checking its absorbance ratio at 260 and 280 nanometer. And if it was found to be about 1.8, then it is considered as pure DNA. So this was the qualitative analysis. Means what is the quality of our DNA? Is it pure? What is and we also measured its concentration. Okay. Now we want to quantify the DNA. We'll quantify our DNA. We'll do quantification of DNA using Agarose gel electrophoresis. So when we want to check the quality and double check its quality, we do use Agarose gel electrophoresis. So now I'll explain the principle of Agarose gel electrophoresis. We all know that our DNA is negatively charged, right? So DNA is negatively charged because of what? Because of phosphate groups in it. Because our DNA do have negatively charged phosphate groups, that's why phosphate groups in it, that's why it is negatively charged. Overall, it exhibits a negative charge. So we'll use electric field. Electric field to have two components, positive and negative. Now we'll use a gel. Gel means a gel matrix. Gel matrix will have it will look like this microscopically. It will have some pores. And as we know the DNA is negatively charged, we'll apply positive field this side and negative field this side. Our DNA will move towards positive side. And because we have created this matrix with small pores in it, the speed of DNA, the affinity of DNA towards positive charge, even if it is a large fragment or small fragment, it will be same. But due to this pore, if the DNA is of small size, it will go rapidly into the pores and it will reach towards the positive end more easily, more quickly than the large fragments, because the large fragments, let me change the color. Because if the fragments of DNA is large, they will not be able to cross these pores, small pores, no, which are formed by the Agarose gel matrix. So they will take some more time, and even it also may be possible that the large fragments just stuck there and won't be able to move. But the small fragments, because they are small in size, so they can easily move and cross through these pores and reach towards the positive end more quickly than the large fragments. So what will happen if we have applied the positive field towards this and negative field towards this? What will happen? The large fragments will remain behind, and the small fragments will migrate more easily. Okay. Um, I just want to tell you one more thing. If our DNA is negatively charged, of course it is negatively charged. Okay, just a minute. Let me first clean it and then I'll explain. Okay. Now, when we will use, I'll talk upon this. When we using agarose gel electrophoresis, we can also like have an basic idea of the masses of DNA fragments. How? I'll explain. What we'll do on our agarose gel, there are different wells, and we will load a control 1 kb ladder, or it depends upon what is the potential size of your isolated DNA. Usually we add 1 kb ladder. What is the composition of this 1 kb ladder? 1 kb ladder have different sizes of DNA fragments. Different sizes of DNA fragments. It is like a controlled fragment. We know, like the companies will provide us like a specific pattern like this, and we'll load the ladder, and when we'll like provide the electric field, but we see a characteristic pattern here. Like, let us suppose I load the ladder here, so I'll see this pattern here, this pattern. So the companies have provided us that this is 10 kb, this is 20 kb, this is 50 kb. Let us suppose whatever it is, 80 kb, this is 100 kb, and 1000 kb, and whatever. They will provide us, like if you see this pattern on your gel, when we load the control 1 kb ladder, we'll see this characteristic pattern, and they have provided us this chart that this fragment corresponds to 200 kb or whatever. Okay. So now if my sample DNA is here, let us suppose this is the fragment of 20 kb, and this is the fragment of 10 kb as per our company uh, guidelines. Now, if I find my fragment here, so it is somehow between 10 and 20. It has migrated less than 10, but more than 20. So I can speculate that its mass is in between 10 to 20. If it seems to be in between 10 and 20, so I can say that it is 15 kb. Okay. Kb, by the way, kilo base pairs. Okay. So the principle is, I have already explained that because the DNA is negatively charged, when we provide the external electric field, our DNA fragments will move, drift towards the positive charge, and their mobility is inversely proportional to their size. Inversely proportional to their size means the larger the fragment, lesser the mobility, means you will see the DNA fragments near the well only, the fragments which are large. Okay. And how we'll compare its masses? By comparing it with the standard ladder, 1 kb ladder, or whatever ladder is there. Okay. So let me first clean this and then we proceed further. Now, I hope that you have gotten the basic idea when we have to use agarose gel electrophoresis whenever we want to check the quality of DNA and want to see its size. then we will use agarose gel electrophoresis. Let's move to the next. Okay. Now I will explain how I did. First of all, the agarose powder is there. Commercially, it is available in the labs. We purchase it from the companies. We'll take agarose gel, agarose powder. So, like if I have to prepare 1% agarose gel, let us suppose I am making 40 ml. So, what is 1% of 40 ml is what? 0.4 gram. What I'll do using weighing balance, I will weigh 0.4 gram of agarose powder and I will load it into this conical flask. Okay. Then for making mixture, I will use 1x TAE buffer. It is a buffer generally used either TAE buffer or TBE buffer. The compositions are easily available on internet. So we'll just mix some like tris base, EDTA, all these things we do make, boric acid, we do mix and we prepare buffer. What is the purpose of making buffer? Buffer will like save our solution from drastic pH changes because we know even like changes in pH may leads to disruption in the hydrogen bonds between the DNA fragments. So it may hinder the optimal experimental conditions. So in order to avoid the drastic pH changes in our solutions, we do use buffers. Okay. So generally we use 1x TAE buffer. So we add the 40 ml, I mean, we make up the volume to 40 ml using TAE buffer. And as this agarose powder is not readily mixed in soluble in TAE buffer, so what we have to do is we have to heat it in the microwave for 60 seconds or 1 minute. Basically, we have to heat and boil it until the mixture seems clear. As you can see, the mixture seems very clear. Initially, when we add the agarose powder in the conical flask and add the TAE buffer, the solution seems to be opaque, little translucent or opaque type. But when we heat and boil it, it looks like transparent solution and clear solution. So when the solution is clear, we let it cool for about 60 seconds or 30, depends. You should, like, you can see if it is bearable to touch. The heat is bearable by your hand. So after that, we will arrange this. We'll arrange this gel matrix apparatus. I can't see my pointer. Okay. So this is this you have to set. There is a tray, gel tray. You will do taping at its four ends. As you can see this color, now this is tape. Okay. And after that, we will load this comb. This is comb. It will help to create the wells, some depressions where we will load our DNA sample. And then we will set it here because the solution don't leak and the base and the four boundaries are just square, and the sharp boundaries should be found, no? So then we'll load this solution here. Okay. And we'll like let it to be solidified. For solidification, it might take 20 to 25 minutes. It varies upon how much concentration of agarose you are using. If you're using higher concentration of agarose, it will take less time. But if you are taking less concentration, it will take more time. If you will take high concentration, it will solidify easily. Now And one more thing, when your DNA sample is of large size, like if you are, separating genomic DNA, then as the size of DNA will be large, so you will use the agarose gel concentration less. Large DNA size, less agarose gel concentration. Let us suppose 0.5% or what, usually we use 1%. But for mitochondrial DNA, as it is small, so all the fragments will relatively go towards the positive end very rapidly. So you want to separate it now based on your size. You want to increase the resolution. So what you will do, you will increase the concentration. You'll use 1.5%. 1.5% more agarose means the pore size will be small. So the DNA fragments will resolve more efficiently. Now, let's move another. Now this. Yeah, it was a comb there. Now, after solidification, we just remove the comb. Now we can see very nice wells here. Okay. Now what we'll do, we'll just remove the tapes. After removing the tapes, we'll just keep it in this tank. This is gel tank. This all is tank. Anode, cathode. One will provide positive and one will provide negative, no? This is anode. This is cathode. Here, this is our gel slab. These are the wells. I have already loaded here. What we'll do, we'll take our DNA sample, like take a paraffin dye, loading dye we'll use. Keep a drop of loading dye using micropipette. Then take the DNA sample, mix it using the pipette. We'll mix it. After mixing, we just aspirate it using the micropipette and then load here. Similarly, we'll load many samples, whichever we want. Here I did loaded only 3 samples, but the rest of all the wells contain dye. Out of these 3 samples, 2 are samples and 1 is control, that 1 kb ladder. Okay, let's move on. Yeah, so this is the electrophoresis. Using this apparatus, we will provide the electric field. So this is red is anode, this is cathode. We'll join these wires to the its respective positions. And also in this tank, we'll load 1x TAE buffer also. TAE or TBA, whatever you are using for making gel, no? First, we'll equilibrate it, and then we will load our samples and keep our slab for at 100 volts for some time. And for how much time we have to run this apparatus? Till our dye just flow down. You won't be able to see the dye. Okay. So now the electric field, we have provided the electric field. Under constant electric field, what will happen? Our the small DNA fragments will migrate towards the positive end. Our large fragments will also migrate towards the positive end. But as the size of larger fragments is more, so the pore size will hinder its movement. So the larger fragments will leave behind, and our smaller fragments will lead. Hence, we'll see this kind of pattern on our gel, and we'll able to resolve our DNA fragments. So after some time, it looks like this. We can see it under UV transilluminator. And one more thing that I forget to tell you. I'll go back and tell you. Here when we added the gel now, we also added uh, 0.1 micromolar or 1 microliter. 0.1 microliter or 1 microliter ETBR. ETBR is a stain. It will like the DNA is like this, na? So it will just stick to the major and minor grooves of the DNA. So when we'll shine UV light on this stained DNA, our DNA bands will look orange. Okay, so I forget to tell that in our gel, we also have added ETBR. ETBR is a stain and it is carcinogenic. We are not supposed to touch it with our naked hands. We usually wear gloves and then we'll suck it using the pipette and load. Okay. Yeah. So after this dye run out, then what we'll do, we'll take out this slab and keep it under UV transilluminator. It is an instrument which will shine UV light on it, no? As we have added ETBR in our gel, so our DNA will be stained using ETBR. Now when we have shined UV light on it, they will look orange. So as I have told you that I have loaded two samples. These are two samples and this is the 1 kb ladder. Now as you can see these light bands here, no? This is 1 kb ladder and these are our samples. This was something I have overexpressed in it, like this. It's basically something what I forget what it is, but yeah, I mean what samples I have put here, I forget. But the thing is that the main thing you should be able to understand that the larger fragments are left behind, but the smaller fragments are migrated towards the positive and more fast than the larger fragments. So this was all about Agarose gel electrophoresis. I will upload an article on our website. So you should go and check that article. Plus the potential interview questions also I'll upload there. So thank you. You can visit our website, cybits.in. And if you are interested, you can join our Telegram group. The links are in the description. Thank you.